Thank you, Arne. That was a generous introduction. You've always been one of my superheroes in the field, so it's very nice to be here and to come back to Stockholm, which I've been to a number of times, and it's always been a pleasure to come here and meet my friends from this area. So, as Arne said, I'm going to talk about fearful brains and what that can tell us about therapy, fearful rat brains in particular. And we'll start with the simple behavior that we use when we study all of this, and it's Pavlovian fear conditioning. So a rat is placed in a small chamber, and the rat hears a sound, and if nothing happens, the rat begins to ignore the sound, but if the sound is paired with an electric shock, and you only have to do this one time, then the rat develops a fear of the sound because it predicts the electric shock, and this becomes a lifelong fear. As long as you present this sound to the rat, again, as long as the rat lives, the rat will be afraid of this sound unless you've done things during the rat's life to weaken that stimulus. But even if you weaken it through, for example, extinction processes, the fear always comes back. The fear is almost more or less permanent and is a characteristic of the animal for the rest of its life in one way or another. So the way these responses are expressed, what we often measure is the freezing behavior of the rats, because many animals freeze when they encounter a sudden danger. And freezing is a good thing to do, especially in an animal that's subject to predation by other animals, because predators tend to respond to movement. So if you remain motionless, you're less likely to be detected by the predator. And also predators will become excited and worked up if you start jumping around. Many animals will play dead, for example, to keep the predator from attacking. So freezing is a pretty good thing to do. It works for most animals most of the time. It doesn't work so well for a deer that's stuck frozen in your headlights because evolution hasn't caught up with that process of fast-moving artificial objects yet. So, but for most animals most of the time, freezing is a pretty good thing to do. There are increases in blood pressure, heart rate, respiration, and other autonomic indicators. And as Arne said, we measured blood pressure in the early days of our research, but decided that for the questions we were interested in, we could answer most of the things by measuring freezing behavior. And sometimes we've also measured stress hormones, but it's important to realize that these also exist. And these are all part and parcel of a defensive response that helps keep the animal alive in any particular situation. So the reason blood pressure and heart rate are going up is not because those are stress responses, but it's part of the response that's required to move blood around in the body to get metabolic energy to those parts of the body that need the energy to deal with the fight-flight response. So a rat that's freezing is in preparation for either having to fight with the predator or to try to escape, and so blood needs to go into its legs and arms for either to escape or to fight, needs to go to the brain, and that blood has to be taken from someplace in order to provide the energy. And it's usually taken from the skin and from the gut and systems like that can postpone their responses until after the fight or the flight is over. Then that's one of the reasons why you feel sort of chilly during a fearful event, because the blood is moved from your skin. It's no longer cooling your skin, and so you have a kind of chill about you when that happens. Well, across a lot of research, the amygdala has turned out to be a very important part of the neural system that is responsible for these kinds of simple responses, these simple fear responses to relatively simple external stimuli. So it doesn't matter whether you use a light, a tone, an odor, a context. It doesn't matter whether you measure freezing, blood pressure, heart rate, or hormones. And it doesn't matter so much what kind of animal you do the experiment in. The amygdala is a key part of the circuitry through which the external stimulus comes into the brain and elicits these fear responses, and also a part of the circuitry through which the fear learning takes place. Now, in a way, it's inappropriate to talk about the amygdala because it's not the entire amygdala that does any of this. It's only a couple of very small pieces. So important are the lateral nucleus and the central nucleus. The basal nucleus may play some role as well, but the main areas are the lateral and the central. The lateral is important because it receives all of the sensory information from the external environment, allowing the animal to detect the danger that's present. The central is important because it connects with all of the autonomic and motor response systems and controls the expression of the various responses. So because the lateral nucleus is this sensory gateway, it receives, in the case of the auditory system, it receives thalamic and cortical auditory inputs. And in the case of the unconditioned stimulus of the somatosensory pain system, it also receives thalamic and cortical sensory inputs. And all of these inputs converge onto individual neurons in the lateral amygdala. So that's why the lateral amygdala is so important, because it's able to integrate the CS and the US through both of the main routes of information flow from the thalamus and the cortex. So there's a tremendous amount of information convergence onto those single neurons in the lateral amygdala. And as a result of the stronger stimulus, the shock, unconditioned stimulus, depolarizing these amygdala neurons, that is, causing them to fire action potentials, at the time when they're receiving weaker inputs from the auditory system, the learning takes place. And we'll come back to this towards the end of the talk, I think. But that's just kind of the basic framework under which we operate. So sometimes you hear about the basolateral amygdala. And to me, this is not a very useful term because it covers way too much information, too much territory. So it includes a lot of different structures, only one of which is the lateral nucleus. And the lateral nucleus is just a small part of it. So this is like trying to tell someone about New York City by talking about New York State. New York State is very different from New York City. And if you want to know about New York City, you need to go to New York City and not to upstate New York. So that's why we focus here on the lateral amygdala, not the other regions of the amygdala. So let me talk a bit about some various kinds of simple things that we can do in rats and have done in rats that have important implications for the treatment of human disorders in various ways, mainly fear and anxiety disorders. So I'll talk a bit about extinction or exposure therapy. I'll talk about something called reconsolidation. Then we'll combine extinction and reconsolidation. We'll consider how you might have extinction without ever arousing fear. And finally, we'll talk about the difference between passive and active coping. So let's start with extinction. So in the early 1990s, Mariah Morgan was a graduate student in my lab, and she was interested in the question of how rats extinguish and what parts of the brain are involved in extinction. Now, extinction is the presentation of a stimulus like the tone that's been paired with the shock. You present that over and over again until the rat stops being afraid. Just as in exposure therapy, you would present the feared stimulus over and over to a human in a safe context and weaken the response so that they're no longer afraid of the stimulus. Well, what Mariah Morgan found was that when she lesioned these areas of the prefrontal cortex, the rats were no longer able to extinguish. Here you can see they continue to extinguish, whereas the control groups exhibited extinction. So we call this emotional perseveration to sort of map it onto the concept of perseveration that Luria and others had talked about in terms of the more lateral parts of the prefrontal cortex. And in their case, they were mainly talking about cognitive perseveration. But we were taking this idea of perseveration into the emotional domain, where in spite of the fact that the stimulus should be weakening, it continued to have a persistent fear-arousing effect, despite the fact that under the conditions the animal was experiencing, it should have lost its fear of this stimulus. So the idea is that perhaps in people who have persistent fear that's difficult to treat, there may be something, I won't say necessarily pathological about their prefrontal cortex, but something different about their prefrontal cortex that makes it hard for them to get over the fear that other people more easily are able to get over. So this was something that we discovered, and then many people have followed this up. In particular, Greg Quirk was in my lab at that time, and when he left the lab, he decided he wanted to pursue extinction, and he's done a beautiful job of carrying out studies of behavior and physiology and pharmacology to try and understand some of the mechanisms of extinction. I'm not going to go into the details of his work, but he's now one of the leaders in the study of extinction. And we know quite a bit now about the role of the medial prefrontal cortex in this kind of extinction behavior. So the basic idea of the circuit that's sort of emerging out of this is, well, here's the lateral amygdala and the central amygdala. So the lateral is getting the external stimulus. The central is controlling the freezing and other responses. And I mentioned the basal nucleus as well. It's connected with both the lateral and the central. And it's getting inputs from the hippocampus, also from the prefrontal, although not shown here. The hippocampus is important because it provides a context in which you decide to be afraid or not. So a snake on a path in front of you in the woods is dangerous, whereas one in a zoo is not dangerous to you. So your brain has to separate out those kinds of situations, and the hippocampus seems to play an important role in that, based on our studies of the effects of hippocampal lesions on context conditioning in rats. But it's this ventromedial prefrontal cortex that seems to be especially important in weakening the amygdala's response or in regulating the amygdala's response. So the amygdala can be thought of as the sort of accelerator for the fear car, and the medial prefrontal cortex is the brake. So if there's something wrong with the brakes, it can't slow down the car because the accelerator keeps being pressed without being able to be stopped. And so the medial prefrontal cortex is a very important region in this. Now, in spite of the fact that exposure therapy is more effective than, say, talk therapy in controlling fear, the problem is that through spontaneous recovery, in other words, just the passage of time, through reinstatement, which is the re-experiencing of the traumatic stimulus, say in the rat's case, a foot shock, or in the case of a human with a phobia, re-exposure to the phobic stimulus, or simply being returned back to the environment in which the initial trauma might have occurred. These are enough to bring back the fear. Stress is particularly important as a stimulus that resets all of this and allows previously extinguished fears to come back. So a person can be successfully treated for, say, fear of heights, but then the patient's mother dies and the fear comes back. So in spite of the fact that exposure is pretty good, it has problems. And here's an example of spontaneous recovery in a rat. So the rat is afraid because he's been conditioned here, and then he's extinguished and the fear comes down, but then the fear recovers simply by the passage of time. Overnight, you get a huge return of fear by doing nothing. So exposure is useful, but it recovers. So people like Mike Davis have been attempting to use various forms of pharmacological treatment, like D-cycloserine, to enhance the effects of extinction so that it doesn't quite come back as strong. And there are a lot of other options, other possibilities that are emerging out of rat and other animal research about what kind of targets might be useful in regulating extinction. I'm not going to go into that because that's not the main thing that I want to talk about today. But we just want to make note that through pharmacological treatment, you might be able to make extinction better. So let's turn to the topic of reconsolidation. So each time you form a memory, your brain synthesizes proteins, and the proteins are synthesized in the neurons that are forming the memory. So those proteins, when activated, then go back to the synapse that's forming the memory and strengthens those synaptic connections. So in the case of simple fear conditioning, where you have the tone and the shock triggers the synthesis of proteins. Those proteins then come back to the synapse and make the CS, the tone, more effective in activating that neuron the next time that the tone is presented. So we've known about the role of protein synthesis in the formation of any kind of memory since the 1960s. So it's been established that if you block protein synthesis after you train an animal on some task, the animal will show short-term memory, but not long-term memory. So the protein synthesis is not necessary for the temporary formation of a memory, but in order for that memory to persist, the protein synthesis has to take place. Now, because the drug is given after training in these studies, the animal has learned the response, so you're not interfering with learning, you're interfering with the conversion of an intact short-term memory into a long-term memory. So, as I said, this has been known since the 1960s, but in 1999, Glenn Schaaf did an experiment in my lab that took it to a new level. What Glenn was able to do was to ask the question of what would happen if we block protein synthesis in the lateral amygdala after training. So because we were able to identify all of the circuitry and show that the lateral amygdala was a key site of plasticity for the acquisition of the fear response and the storage of the fear memory in this form of simple implicit learning, we were able to put the protein synthesis inhibitor directly in the lateral amygdala and show that short-term memory was intact and long-term memory was impaired. All of the previous research on protein synthesis inhibition had involved administration of the drug systemically. So this makes animals sick, and you don't know whether they're not responding because they just aren't motivated to give you the answer that they may know, or whether they actually have forgotten or are unable to form the long-term memory. But when you put a tiny amount of protein synthesis protein synthesis inhibitor directly in the lateral amygdala, you're not affecting any of these other motivational processes or drive processes that are necessary to perform the response. You're simply inactivating the ability of the neurons to form a new memory through protein synthesis in that small part of the brain. And when you do that, again, you see short-term memory is intact and long-term memory is impaired. So you need protein synthesis in the lateral amygdala to create a fear memory. Now, Kareem Nader, who was a particularly interesting and bright postdoc, said, well, what would happen if we then block protein synthesis after the retrieval of a memory? Because again, as with the initial protein synthesis findings, it had been known since the 1960s that you could disrupt memory by doing various things after the formation of the memory, and in fact, even after the retrieval of a completely formed, completely consolidated memory. But this research didn't sort of get much traction back then, and it sort of didn't really go anywhere. Susan Sarah did some more studies in the 1990s, but again, it still didn't sort of go anywhere. Where it really took off was after Kareem Nader's study published in Nature in 2000, the year 2000, because again, he was able to show that if he blocked protein synthesis after the retrieval of a long-term memory, a fully consolidated memory, at the site where the memory is formed and stored in the brain, he was able to disrupt the survival of the memory. So just as you find for consolidation, where short-term memory is intact and long-term memory is impaired, when you block protein synthesis after the formation of the memory, when you block protein synthesis after the retrieval of a fully consolidated memory, post-retrieval short-term memory is intact, but post-retrieval long-term memory is impaired. So you not only need protein synthesis when you form a memory, but also every time you use a memory, you have to restore it through protein synthesis. So what's going on there? Why do you need to store a memory through protein synthesis simply because you've used it? Well, it's an opportunity to update memory. Each time you take a memory out, the memory is now subject to change. You add new information to it, you may take some information out, you may form a completely different memory. So you meet me in the bar tonight, you find out something about me. That may change the memory you have of me as a result of my lecture here today. So you take the memory out, it becomes unstable, that allows you to update it or change it, and then you have to store it because now it's a new memory. So that's the idea under the protein synthesis involvement in reconsolidation. Now this is a very specific process. So here's an experiment that Jacek Debish, Valerie Doyer, Marie Monfils, and Glenn Schaaf did. They're using two conditioned stimuli, CSA and CSB. These are conditioned at the same time. Why did they do that? Well, if you have one stimulus paired with a shock, that's a very simple thing. But imagine a human memory. It's a very complicated thing with lots of different things associated with lots of other things. And while we haven't necessarily fully modeled a completely complex human memory, what we've tried to do is make it more complex by adding components to it. And this is just one example of how we've done that. So the rat is conditioned to these two stimuli with the same US. So a high tone predicts the shock, and a low tone predicts the shock as well. It all happens at the same time. They're conditioned in the same chamber. And then later, either the high tone or the low tone is presented, and paired with a drug that blocks the reconsolidation of the memory. And then both stimuli are then tested. So again, they're conditioned with 2 tones. One of the tones is reactivated. Both tones are tested in short-term memory, and then both tones are tested in long-term memory. Only the tone that was reactivated in the presence of the drug is affected. So that means that even though we've created a slightly more complex memory, when we give the drug in the presence of that stimulus, we don't affect the entire memory. We only affect the component of the memory that was explicitly active at the time of the retrieval. This is important because one of the criticisms of reconsolidation research, as you take it into therapeutic settings, is that, how do you know you're not going to erase someone's personality if you erase a traumatic memory? Say, let's say you have a person who was a Holocaust victim after World War II, and this became a big part of their personality, and they lived for 50, 60 years with these memories, and now you're able to somehow dampen the troubling aspects of that memory through these kinds of procedures. Would you then erase their whole personality, which had been structured around those memories? And the answer is no. The patient and therapist could slowly and selectively sort of chip away at the memory until they were comfortable with what they had removed. Now, the other aspect of this, though, is that from the studies that have been done so far in humans, the explicit declarative memory is not so much affected. What seems to be affected primarily is the more implicit emotional aspects of the memory that are mediated by the amygdala. So the autonomic arousal, the muscle tension, and so forth. Not your memory that you were in the concentration camp, but the troubling anxiety that's triggered every time you remember that is dampened. So that's the hope for this kind of research, that will be the ultimate outcome, and that's the way things are going now. That's not to say that reconsolidation does not occur for declarative memory, but when it's part of an emotional memory like this, it seems to target the emotional component particularly well. So the basic idea is that, the traditional view of memory is that when you retrieve a memory, you retrieve what you store the first time, the original time. So you form a memory, and then every time you remember it, you're taking the same thing out. But the reconsolidation view is that when you form a memory and you store it, and then you retrieve it, the next time you retrieve what you stored the last time rather than what you retrieved the first time. So your memory is only as good as your last memory in this model. This is the most extreme version of the reconsolidation hypothesis, but it's very consistent with a lot of what we know about memory. For example, a person who witnesses a crime and then goes to court to testify, but tells the court about what they read in the newspaper rather than what they witnessed on the day of the crime. So reading the newspaper reactivated the memory. That gets stored in their mind as part of the memory, part of what they experienced on the day of the memory. In fact, they never really experienced this. I mean, Dan Schachter has done many studies at Harvard on the creation of false memories, and people are very, very committed to their false memories, even though they never experienced these kinds of things. So whenever you take a memory out, it gets changed, or it has the opportunity to be changed because it becomes unstable as a result of the retrieval process. And this lasts about 4 or 5 hours, this instability. If you block protein synthesis 6 or 7 hours after the retrieval of the memory, it has no effect. You have to block protein synthesis within that 4 or 5 hour period. That's called the reconsolidation window, and that'll be important in the next thing that we're going to talk about. But this has become important as a tool for treating post-traumatic stress disorder, and there have been some pilot studies that have shown some success, but it's hard to get enough patients to do these studies, patients who aren't on other drugs that make it difficult to examine the effects and so forth. So there isn't a clean, very convincing study yet in humans that shows that this can be used to treat post-traumatic stress disorder, but a lot of studies are underway. In rats, it's also useful in the treatment of addiction. For example, rats that are addicted to cocaine and then given a cue that's associated with cocaine will relapse and take the drug again. But if they're presented with the relapse cue and then reconsolidation is blocked, when they get the relapse cue later, they don't relapse. So this is a potentially powerful technique that can be used in a lot of different situations. And I think that's the work of Barry Everett at Cambridge on the drug relapse study. Okay, now we talked about reconsolidation and we talked about extinction. So Marie Monfield had the idea of combining these two processes. We know that, as we said earlier, extinction is simple. It's just a behavioral manipulation, which is good. You don't need drugs to do it. But it's temporary and weak. It bounces back. Reconsolidation seems to have a more lasting effect, but it requires the use of drugs. Maybe these two can be combined. Maybe these two ideas can be combined somehow. So her idea was, instead of giving a drug, what you do is you train the animal, and then after the memory is consolidated, you give a retrieval trial to destabilize the memory, as in a reconsolidation experiment. And while that memory is unstable, you teach the rat that it's safe. The tone is now safe because nothing happens. You do extinction during the reconsolidation window, that 4 or 5 hour period. And then you test long-term memory later, and here's what she found. When she conditions the animals, waits 24 hours. They either have no retrieval or they have a retrieval trial. The retrieval trial is either 10 minutes, 1 hour, 6 hours, or 24 hours before the start of extinction. So if she does the extinction 10 minutes or 1 hour after the retrieval trial, there's no spontaneous recovery. If she does it 6 hours or 24 hours later, there is spontaneous recovery. So you have to be within the reconsolidation window when you do this. Now, you may be asking yourself, what's the difference between simply doing extinction and doing extinction with a retrieval trial? Because the first trial of extinction is a retrieval trial. The key thing is you have to insert a break of at least 10 minutes in there to trigger the reconsolidation process and allow those mechanisms to take off. Once that 10-minute window has happened and you do the retrieval trial, the brain can then benefit from this reconsolidation process. You then train the animal on extinction, and what the animal walks away with is that the stimulus is safe and not harmful. And it's relatively permanent, much more permanent than extinction itself, because you don't get spontaneous recovery. So Daniela Schiller and Liz Phelps and I and other colleagues picked up on this and turned it into a human experiment as well. And basically they found the same thing, that 10 minutes and 10 minutes so that the fear is acquired, the extinction is done, and then you look at recovery at 10, whether there's a 10-minute, a 6-hour, or no reminder situation. No reminder, they have spontaneous recovery. If there's a 6-hour period after the start of extinction, they have spontaneous recovery. But if there's a 10-minute period after the retrieval trial and the start of extinction, they don't recover. Now, the editors, this was published in Science, and the editors of Science required that they wait 1 year and retest the people to make sure that there was no spontaneous recovery. And indeed, there was no spontaneous recovery after a full year. So this is a potentially very powerful way of treating fear in a way that doesn't come back quite as easily as if you do simple extinction or exposure. And this is a neurological correlate of this. You can see that there are neural changes in the brain as a result of being reminded or not reminded during this retrieval trial. And we won't go into the details of that, but Liz will probably talk about that in her lecture tomorrow. Okay, let's just briefly talk about extinction without fear. So one of the problems with simply doing exposure therapy is that sometimes when you expose people to their fears, you make things worse, because each exposure is a retrieval of that fearful memory, which releases norepinephrine and other things in the brain, and activates the emotional memory systems, and causes a fearful state. And each retrieval can then sort of feed forward onto itself so that you have more emotional arousal rather than less. So often when you try to reduce the fear, you first get greater fear that then has to be extinguished. And people obviously don't like this. They don't want to be exposed to their fear-arousing stimulus. As you probably know, people with anxiety disorders will do anything to avoid reactivating their fear. So they're extremely good at avoiding situations that are going to activate the fear, which means they never get the opportunity to naturally extinguish. So here's an example in rats of extinction without fear. This is not an experiment we designed. It's simply an experiment that Kiriana Cowensage, who was a graduate student at the time, sort of stumbled across as she was doing something else. So basically what she does is she puts the rat in the box, and when she puts, in this case, they're in the box and there's no sound on, and then at some point later the sound comes on. Then they freeze. But she puts them in the box and the sound is already on. When she tests them later, they don't freeze at all. So so here's an example. So they're trained and they all show the fear response. And now they're returned to the test chamber. And in one case, the tone is not on. And so when the tone comes on, they show a lot of freezing. If they go in with the tone already on, they never show any freezing. Both groups extinguish very well. So by the end of the session, they've both extinguished. The question is, do these rats that are extinguished without ever expressing fear then show extinction later? Or are they simply ignoring the sound altogether? And what you see here is that both groups showed the same amount of extinction and spontaneous recovery and so forth. So you don't need to actually express the fear in order to weaken it. You simply have to be exposed to the stimulus to weaken it. Now, we don't exactly know how to turn this into a therapeutic procedure at this point, but it does show that there is the potential for having exposure and weakening fear without ever experiencing the fear itself. Now, this kind of passive exposure to stimuli, while it's useful, has proven not to be effective, as I said. A much more effective way of treating fear is to have the patient or the rat, in either case, be actively involved in the process of reducing their own fear. So this is a picture from Wall Street, not the movie, the place, in 2008 when the stock market was crashing. So this guy is watching, the numbers go down, 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 down. He's having his fear response. What's he going to do? Well, he's going to run up to his office and take some action. So first he reacts, like we all do in a fearful situation. We react, but then we can't react forever. A rat that's frozen in fear in the face of a cat has to do something at some point, either run into a hole, run away from the cat, fight with the cat. So we became very interested in this transition from reaction to action and how people become emotional actors. What goes on in the brain as you make that switch from a passive reaction, which is an involuntary response that just happens to you, to an active response where you voluntarily are in control of your fear. So we studied this using a very old technique, avoidance conditioning, which in the 1940s and 50s was the primary means by which fear was studied in the brain. In the heyday of behaviorism, instrumental conditioning was thought to be more like human behavior than Pavlovian conditioning. So researchers are much more interested in studying instrumental responses. The problem is that they didn't know enough about the brain, and this was a very complicated task to go in and try to understand basically from scratch. But now we know that in avoidance conditioning, the first phase of avoidance conditioning is Pavlovian fear conditioning, and then that transitions into instrumental avoidance learning. So now that we understand the circuitry of Pavlovian fear conditioning, maybe we can build on that to understand how avoidance works. So that's what we're doing in these studies. So this is a signaled active avoidance task. So there's a tone, and the rat is in the chamber, he hears the tone, and then there's a shock. So it's basically Pavlovian conditioning right there. And what he has to learn is that when the tone comes on, he should move to the other side of the chamber to prevent the shock from coming on. So the beauty of this task is the stimuli are identical to what we do in Pavlovian conditioning. It involves a Pavlovian conditioning component, but then the rat has to learn to do something. So the CS in this case starts out as a Pavlovian elicitor of fear, but then it becomes an incentive stimulus that the animal is using to motivate an instrumental response that will reduce the fear that the warning signal warns about. It also becomes a second-order conditioned reinforcer, because over time, the rat will learn to perform this response simply to turn the tone off without ever experiencing the shock. So it's an interesting task. These are interesting tasks to explore, and the behaviors that result are also very interesting. So here's the acquisition of avoidance, the instrumental active behavior. And what you see is it mirrors the extinction of fear. So this is freezing going down. This is active avoidance going up. You can't take action if you're frozen in fear. So it's sort of automatic that you have to stop freezing in order to learn active avoidance. But keep that in mind because it'll come back to be important in a few minutes. Now, rather than the lateral and central amygdala being important in avoidance conditioning the way they are in Pavlovian fear conditioning, it's the lateral and the basal amygdala. Central nucleus lesions have no effect. So the CS and US converge here in the lateral amygdala in both cases. That CS is then used to motivate and reinforce this instrumental action by way of connections to the basal amygdala. But in order to do that, the connection from the lateral to the central amygdala that controls these involuntary fear reactions, somehow this has to be suppressed. And that's an interesting question as to exactly how that comes about. Now, how do we know that that has to be suppressed? I'll come back to that in a second. So what we decided to do is to lesion the central nucleus of the amygdala in a group of rats that never acquired the avoidance response. So in every experiment, about 20 or 30% of the rats simply can't avoid. Usually they're just thrown out, and because they can't learn the task. So here they are. They've gone through 3 sessions showing no signs of avoidance at all. So we said maybe when we lesion the central amygdala, they'll learn the avoidance response. To our surprise, they had already learned it, but they were so frozen in fear they couldn't express this avoidance response. They did learn it even better, but the important thing was they knew the answer, but they couldn't get it out. Somehow, therapeutically, we have to figure out how to relieve this pressure exerted by the central nucleus that causes people to be stuck in this passive mode of coping and unable to achieve an active mode where they're in charge of their fear. And the reason that, the reason it's important to do the active coping, if I can get the slide, is this. This is a complicated slide, and I'm not going to go through the details, but let me just tell you what it means. So in this study, one group of animals was in charge of their fear. They learned to perform behaviors to prevent the CS from ever coming on, and therefore prevent the US from coming on. These animals, when tested later to the CS, never show spontaneous recovery. Another group of animals had the exact same stimulus presentations. They were what's called yoked animals. They get the exact CS, the exact US throughout the experiment, but they never have control over it. These animals do show spontaneous recovery. So passive coping with the fear-arousing stimulus will reduce it, but it'll come back. But if you're engaged in active coping, you're much less likely to get spontaneous recovery. So Bezel van der Kolk, who's a PTSD therapist at Harvard, has developed a treatment for rape victims on the basis of research like this. And what the patients do, what the victims do, is they have to go through physical fights with intruders. This is not, I mean, it's role playing, but it's not just kind of like joking around. This is very serious, intense fighting. And he finds that this engagement of the body in the therapeutic process, this active instrumental coping, is much more effective than simply passive talking or other forms of therapy that are used more traditionally. So there's something about this active engagement of the physical body in the coping process that makes the treatment much more effective. And that's the same thing that's happening with the rats. If they're actively and personally engaged in reducing their fear, then they do much better on the later when they have to face their fears. They don't spontaneously recover. So this puts it all into a bigger circuitry. So here's the initial circuit, lateral and central amygdala. Basal amygdala is out here. Central amygdala controls the reactions. Basal amygdala communicates with the ventral striatum. If you block this connection, you don't get these actions. Ventral striatum allows you to enter into corticostriatal motor circuits. These circuits are modulated. Dopamine probably plays a big role over here. We think norepinephrine and serotonin are more important over here, although in both cases they play some role. It's more about the balance. And so what we tried to do is start from a very simple kind of behavior with a tone and a shock, expand that out into more complex behaviors so that now we're engaging more systems of the brain and sort of trying to just build up a more complex fear network from the bottom rather than with some pre-existing top-heavy concept like the limbic system concept, which tells you what parts of the brain should be involved in emotion. What we're saying is let's forget the limbic system and try to understand how emotion systems work empirically by building the system on the basis of studies of specific behaviors like this. So I'm going to just briefly say that one of the things we've also been doing is studying individual differences because, each of us is unique. As we saw, some rats learn avoidance, others don't. And so we've tried to understand a bit about how these individual differences come about. We can simply take a group of normal rats and condition them, and some will be unafraid and some will be very afraid. So what we want to do is go in and try to figure out, what are there structural features of the neurons in the amygdala that make these unafraid versus these very fearful rats different? Can we do gene profiling in the lateral amygdala and see whether there are genes that are activated here and not in these guys and so forth? These are, at this point, we don't have any data, but this is something we're trying to do. And I just want to, let's see if I have time to briefly tell you about some new results. What we've been trying to do is to mimic fear conditioning by using direct optical activation of amygdala cells. So this allows you to study the effects of, as I mentioned earlier, direct depolarization of amygdala neurons by the optical stimulus. You can see the cells are activated here with CFOS, or you can turn on the light and you get action potentials throughout the light. The light activates this channel rhodopsin, which has been put into the cells by a virus that was injected into the amygdala earlier. And when you shine the light in there, it causes a depolarization of those neurons on a millisecond by millisecond level, as long as the light is on. So it gives you very precise control over the activity. And what you can do is when you pair a real CS, a tone, with a direct depolarization of these lateral amygdala cells, you get fear conditioning. And I'm not going to go through the rest of these details because it's getting late here, but this is sensitive to norepinephrine because if you block propranolol, the conditioning is reversed, basically. So we think that norepinephrine, the neuromodulator, is modulating Hebbian plasticity. Hebbian plasticity occurs when the weak CS is potentiated by the strong US, and then the release of norepinephrine onto that adds a boost to that and makes the conditioning stronger and more persistent. That was work that Josh Johansson published this past year. This is serial electron microscopic reconstructions of neurons in the lateral amygdala that Linnea Ostroff has recently done. So these are real data. So here's a presynaptic terminal. These are the vesicles. All of this, again, reconstructed in very tiny sections through the lateral amygdala. Here's the postsynaptic spine, mitochondria in the blue, and the smooth ER here, and the spine apparatus in the orange. So why did she do this? Well, what she found was that when she conditioned rats to be afraid versus to learn that the stimulus was safe, where there's no shock, she found that there were bidirectional changes. So very small spines increased the size of the postsynaptic density in fear conditioning and decreased it in safety training. So these are structural morphological characteristics that we can identify in the lateral amygdala that occur as a result of fear conditioning. And I'll skip some of this other detail here. These are a lot of people that participated in these studies, and I don't have time to go through all the details. But I want to close. You saw a picture of my band at the beginning, and I just want to close with a music video. It has an educational component, so you'll see. It's based on an Emily Dickinson poem called Fearing, and the music in here is original music that our band writes, and most of our songs we write the lyrics to as well. And this will also feature Daniela Schiller, who was a student, postdoc with Liz Phelps and me. Daniela is now a professor at Mount Sinai. So if we can have the lights down a little bit and see if the video will start. Lights down more.
While I was fearing it came, but with less of the fear, because fearing it so long had almost made it dear. There is a fitting dismay, an appropriate despair. 'Tis harder knowing that fear is due than knowing it is here. The waiting is the worst. It ties you up in knots. Anticipation a curse. 1,000 empty shots. Fear is the most primitive and basic emotion. It occurs when we encounter danger. An animal can put off the good stuff, eating, drinking, sex for days. But responding to danger must be immediate, or there will be no more eating, drinking, or sex. The fear response is the same in humans and other mammals. Muscles tense, heart beats fast, hormones flow. These responses help keep us alive when threats arise. If to fear were merry and to worry were gay, how blithe would be the memory of that awful day when hell was turned loose, a full psychic assault, a fearful memory so cruel. Could it be my fault if recollecting or forgetting, then I remember not? If forgetting, recollecting, how nearly I forgot. But recollecting's not forgetting, it's vivid rehearsal of pain. It reminds me of that day. It keeps fear in my brain. Evolution says if it ain't broke, don't fix it. Indeed, the brain mechanisms of fear are highly conserved. The amygdala is a key structure. It detects danger and produces hardwired protective responses. The amygdala also forms emotional memories. It uses these to predict harm in the future. Fear learning is rapid and persistent. While fear memories can be controlled, they are hard to eliminate. The amygdala is hyperactive in many psychiatric conditions, some of which can be treated with drugs. Others respond better to psychotherapy. We need better treatments for fear.